following for some time here in Denmark. Jonas, you work at the Uppsala University, and you have specialized in clinical neuroscience and analytical chemistry. So you are a researcher. You're also director of the ME-CFS Collaboration at Uppsala University under the Open Medicine Foundation, which is a private worldwide research organization combining many countries in the ME-CFS research. In ME-CFS, you have published a number of research papers, which can be found on your homepage. This will all come out afterwards in our members magazine. And your focus has been to develop tools for screening and develop biomarkers for different diseases amongst ME-CFS. And especially ME-CFS, where you are looking at autoantibodies and analyzing what goes on in the ME-CFS body. And today you will focus your talk on the ME research you and your team have been doing. And after a break, halfway, you will talk about the current post-COVID studies you are doing too. And we have quite a lot of patients now in the ME Association who are diagnosed with ME after Corona. And there are some members also from the newly founded post-Corona organization in Denmark. So this talk will be recorded, so the patients and members who are unable to join today because they are too ill, they will have the opportunity to watch later and to listen also to the questions and answers. So Lina just laid out the program, and there will be plenty of time to relax between speech. So I'll give the word to you, Jonas.
Yeah, thank you, Katrine. I hope you can hear me well. I'm actually not so far away from Denmark. I'm in Lund currently. I didn't realize I was supposed to be at a meeting in Lund to present something tomorrow, so I arrived here. So I'm currently now sitting in a basement at the hotel, so I hope you can hear me well.
You're fine.
Yeah, I'll do this in English, but my Danish is not so bad. I was taking my basic studies in Lund, so in Skåne, so I can understand Danish well. But I guess for your sake, I will stay into English now.
Thank you.
Yeah, and I'm trying to share my slides with you, and as we said, we will have possibilities to ask questions later on. So I hope that is going to be fine with all of you as we see this one. So what I'm going to do, I'm just going to give you a brief overview of what we are doing currently in Uppsala and also in our research centers around the world, and what we are aiming for right now and where we're going. And in the end, I also want to give something about our post-COVID research that is currently going on, and I will explain a bit later why we have taken that on also on the side of ME-CSF. Yeah, as Katrine presented very nicely, I'm currently the director of the ME-CFS Research Center at Uppsala University, and I have my professorship in Uppsala. But besides that, I'm also professor in pathology in US and precision medicine in China in Binzu Medical University. And since not so many weeks ago, I was also installed as professor in veterinary medicine, which may be a bit odd to some of you, but it's because we can use some of our technologies and development in biomarkers for veterinary medicine also. So that's part of my interest to broaden our knowledge, basically. This issue with post-infectious consequences and its role for disturbances in neurocognition has become extremely important, not only to all patients and relatives and family members who have members in the family that has been hit by this issue, but actually to the common public in Sweden and in the rest of the world. So this slide just show you from the Swedish television Foredrag that I talked about, that was a remarkable interest from patients and the public audience to listen to what is going on in the central nervous system when it comes to post-infectious diseases and neurocognition specifically. But here is the slide that I wanted to show you, where we sum up the knowledge we have about myalgic encephalomyelitis or chronic fatigue syndrome. We typically in Sweden now has turned into call it ME instead of the full name, or just leave chronic fatigue syndrome out, just to make it a bit more easy for patients and relatives and family members to remember what it is. So, as I said, ICD-10 G9332 stands for multiple system neurological diseases with often a post-viral startup or consequence. And we still don't know exactly why you get ME. As you know, we know that there are risk factors, there are the infection in itself, the genetic and environmental factors that sums up and make a person more vulnerable to have a disease like ME. And when we now, well, as I'm going to talk about later today, when we reach the post-viral, post-COVID group, we see the similar type of genetic and environmental factors that can explain why some people get long-term consequences of post-COVID. Yeah, some of you may know this, of course. Or ME CFS has most common symptoms: post-exertional malaise, PEM. So that is a key hallmark feature that we are looking for in our patients. Symptoms worsen after exertion, persistent fatigue, sleep disturbances, pain, central pain and headache, or peripheral pain and muscle pain. Neurological and cognitive symptoms: brain fog, confusion, disorientation, difficulty concentration, short-term memory issues, ataxia, and muscle weakness, hypersensitivity to noise and light. And this is things you recognize, of course. And then on top of this, in this multi-system disorder, we have effects on autonomic and immune system, and also in the digestive tract symptoms. So orthostatic intolerance, POTS, flu-like symptoms, problem with temperature control, feeling sick from different areas of the body like sore throat, tender lymph nodes, and then problem with change in body weight, and also problem with your digestive system, irritable bowel syndrome, etc. So a very, very complex disease picture, as you know. And the number of patients, this is an increasing number now with post-COVID also, but we say approximately 20 million patients with ME-CFS. In US, that would be over 2.5 million patients, and in Sweden, if we're careful, we say about 20,000 patients, but probably it could be over 40,000 patients in Sweden. Most of the patients, 50% of the patients, can keep a rather ordinary situation, lifestyle situation, if they are very careful with their energy, so they can maintain, for instance, a half-time occupation or study in school, maybe full-time or half-time, but they have to spend most of the rest of the time during the day to rest and store energy, basically, for being able to cope with the situation. 25% are sort of doing quite well, although they have effect on their daily behavior. They can still work and maintain studies in almost 100%. But the worst case patients are, as you know, those that are about 25% that are fully bed-bound or home-bound at least, and they have a very difficult situation. And in Sweden, many of them are isolated and also don't get the support that they need from healthcare and from the government when it comes to economy and so on. So ME-CFS is actually one of the most common chronic illnesses. It's twice as common as multiple sclerosis. And multiple sclerosis, if you ask anybody, basically in the population, they have heard about MS or multiple sclerosis. They know a bit what it is. But if you ask about ME, still there is a lack of knowledge and understanding. And as we all know, ME strikes anybody, men, women, children, any time in life, and so on. Even if we know that there is an overrepresentation of about 60-70% of women, and we know that there are two age groups that are more affected, typically in women, that is late teenage and then pre-post menopause time span. So one can ask, is this a new disorder? Well, ME-CFS in itself has been recorded and reported on for at least 50 years now. But if we look back in history, we see that there is actually plenty of outbreaks of epidemics of myalgic encephalomyelitis character. So maybe not ME, but very similar in symptom picture. And this long list of outbreaks is just a subset of all the outbreaks recorded. And I will talk a bit about the Akureyri disease. Happened in 1948, 1949. But some of you also heard about, for instance, the outbreak at the Royal Free Hospital in London or Incline Village in 1984. And if you look in the lower end of the table, you also see Bergen. There was an infectious disease outbreak in the water. It was Giardia infection that led to outbreak of ME. And now we have the coronavirus pandemic that, well, still is going on, I would say, but with maybe less intensity. So in Akureyri, up on Iceland, the north part of Iceland, there was this outbreak in 1948, 1949 with in total actually over 1,000 patients involved in that outbreak. And it was in the locations in the red circles, as you see on the map here. And in 1955-56, there was another outbreak in the blue circles, 314 cases then. And I had earlier this year, I had the chance to go and visit Iceland and also visit Akureyri. And I visited the junior college and dormitory where this outbreak actually started. And the students that were living at this dormitory, they were affected during a couple of weeks. So there was really an outbreak of an infection. And since samples were not collected from the patients at that time and stored, we don't know exactly what the outbreak was. It was not polio. It was not any of the other known viruses at that time. Most likely an Epstein-Barr virus outbreak, or mononucleosis, as we say. So me and the colleague Fridbjörn Sigurdsson was visiting the dormitory, and we were let in, and I could actually climb up onto the attics, and there was one of the rooms, the dormitory rooms, were actually saved from that time when the outbreak was. So that was a great opportunity to see how the students were living at that time, and they were apparently isolated then in this dormitory during the outbreak, and had to more or less cope with the situation themselves, because healthcare staff and others were afraid that it was a new polio outbreak actually initially. And I also had the chance to meet one of the patients, Asgeir Johansson, who was one of the and still surviving patients at the outbreak. And he actually described how everything happened, how they were, well, of course, very sick and isolated in this dormitory, and did not really get so much support from healthcare at that time. And it took a few weeks before most of them actually recovered. Asgeir himself, he had the symptoms, and he has still symptoms from that, although that's over 90, almost 90 years from, or sorry, 70 years from now, from the outbreak. And it has really affected his life, at least, definitely. About 20% of all the patients at that time had remaining symptoms, and that lasted more than 6 months, and about 10% of the patients had symptoms that lasted for many years, and some got chronic symptoms, as Gail did. So what do we know today? Well, as I said, in most cases, 67% of the reported ME cases are starts with a post-viral fatigue event, and quite often then Epstein-Barr virus, could also be a herpes virus, and some other virus types. It's more common in women than in men. 2/3 of the reported patients are women. It's demonstrated changes in the central and autonomic nervous system. I will get back to that a bit. We know that the metabolism, and especially the energy metabolism, is affected in the patients. And we know that there is an altered immune phenotype and function. So the immune cell profile in ME patients differs slightly from healthy controls. Then there are ongoing research and reports on a changed gut microbiome, small bacteria inside our intestines, and they are affected in many of the patients. I put a question mark there because I don't think still that we have proven that this is a root cause of ME. It could be a side effect of the disease, or being bedbound, for instance, that would affect your gut microbiome. Reports have come out also recently about gene expression, different changes and epigenetic changes in patients with ME, and I think that is very interesting to follow up on. Still need to be proven, I think, in a larger cohorts, and follow up on. Unfortunately, there's a lot of things we don't know yet. So, for instance, what is the true mechanism that causes the disease symptoms, and what leads to the changes we see in so many different organs and symptoms that are spread all over the body, and you don't give symptoms only in one part of the body, like the central nervous system. It affects so many other parts. What are the links that make these changes, and what is the initial triggers? We are sort of understanding now that the viral infection could be the reason for this outbreak of a disease, but is it necessarily one type of virus, or is it any kind of virus? We don't know for sure yet. So, of course, in the end of the day, we want to find out the explanatory model of the disease outbreak, and that is needed for us to know in order to make the selection of the treatments that we would like to use for helping patients, and maybe curing patients also, which is the end goal, of course, not only helping with symptoms. So our specific research focus right now is very much based on high definition, high resolution mass spectrometry. That's a technology that can monitor all biomolecules in the human body based on their molecular weight. And this instrument we can use for doing molecular diagnosis. So when we work with proteins, we do analysis of proteome. That is the full protein complexity in the human body. And from our 25,000 genes, we can produce over 1 million different proteins. So there's a lot of biomarkers to study in our human bodies. We can also study small molecules, and then we call it metabolomics. And the metabolome is making up our phenotype, and even more complex than the proteome, because metabolites can be of any kind of structure, and it can contain almost any element. Proteome and proteins are built by amino acids. So in principle, they are quite simple to study, while the metabolome is more complex. And the proteome we have based on our genes, but we change our proteome over and over again during lifetime, and even within short time spans. The metabolome changes extremely rapidly, so anything we do at any time point will change our metabolome. It's not only genetic or epigenetic factors and age and environment, but diseases, things we eat like drugs or nutrition, and our current lifestyle that will change our metabolome quickly. So how do we measure it? Well, we take samples of different kinds. We typically use body fluids like blood, urine, or cerebrospinal fluid, the fluid that is surrounding our central nervous system. And we isolate the proteins or metabolites, and we then separate them on high-resolution water or liquid chromatography, and then connect that to our instrument, the mass spectrometer, that can actually measure and detect and identify all these molecules in an intricate way. So our central focus currently is to study the central nervous system and focus on the effect on neurocognition, as I mentioned in the beginning. So disturbed neurocognition as brain fog and disturbances in mind, in the memory, in other brain functions, headache, etc., is a central part of what we are focusing on. And we know that the central nervous system, the brain, is typically rather quiet when it comes to inflammation, and that is how it should be, because small inflammations or major inflammations in the central nervous system will lead to bad consequences, scar tissue formation, etc. But sometimes we get activation of infiltrating cells that penetrates the blood-brain barrier, or activation of our resting immune cells in the central nervous system, called astrocytes or microglia cells. And when these cells are activated, they change their shape, and then they start producing inflammatory factors like cytokines that affects the central nervous system in a very potent way. So what can happen is, if we are unlucky, we get an inflammatory reaction signal, we get an activation of our microglia cells, and they start to attack healthy neurons and degenerate the neurons, activate antibodies against neurons, and break neurons down. So we lose neurotransmission or signaling in the brain by the inflammatory process. And these things we can actually now monitor, so we know that this is happening in the central nervous system, and we do it by monitoring the liquid that surrounds the brain. It's produced here in the central part of the brain, called corridus plexus, and then circulates around the brain down into the spinal cord region, where we can do a lumbar puncture and get the volume out to measure the chemical properties of the liquid around the brain. And here's just a small recording of how the brain is looking during an MRI scan. And in this picture here, you see that we have an enhanced movement, and that's actually part of the circulation of the blood pressure variability you get during your heartbeat. And that actually helps the circulation of the liquid that is produced in the central part of the brain, and then circulates around the brain down to the spinal cord region. And we get the sample through a lumbar puncture, where you penetrate between the spines into a region of the lower part of your lumbar region. So it's called the dorsal sac, where we can extract this liquid. And of course, it's a bit invasive, so if we could take a simple test, a blood sample or a urine test to monitor the central nervous system, that would have been fantastic. And we can, in some cases, actually do that, but in most cases, we need to get a fluid that is more close to the organ that we're looking at, and then cerebrospinal fluid is optimal for us. So we have studied this liquid in many different patient groups for a long time, and this is our first paper we published in 2011, where we actually looked at the content of this liquid and compared ME patients, chronic fatigue patients, with patients that had a tick-borne encephalitis or Lyme disease disorder. And we could at that time already tell that there was an inflammatory reaction in both patient groups, but we could also see that there were differences between the chronic fatigue syndrome or ME patient group and the tick-borne infection disease patient group. And together with the same colleagues, basically, we have just now got a paper accepted in Annals in Medicine, where we have compared patients with ME and patients with ME together with fibromyalgia, so with a comorbidity of fibromyalgia. And what we see here is that the ME inflammatory reaction is overwhelming, so it's actually taking over the small signals we could see from fibromyalgia, which mostly comes in the peripheral part of the body, as you know. But we see that the ME central nervous system inflammation is rather potent and strong. We are also now doing a study and following up on preliminary results we had from early studies on ME patients. So this is in a collaboration with the clinic in Stockholm, Braget Clinic. And here we have collected cerebrospinal fluid, blood samples, other healthcare or health inquiries, and we have done neurophysiological examinations, etc. We also collected saliva, as it says here, measured the biomarker content of these fluids, and we also studied the intracranial pressure in patients. And the picture here is the head from underneath, and you can see that here's the eyes of the patient, here's the nose. And what we can do with a rather simple ultrasound technique, we can measure the nerves from the optic nerve, the diameter of the optic nerve, and we can actually tell if there's an overpressure in the central nervous system in the patients. Because if you have a pressure, you compress the diameter of these nerves from the eye. And we can actually see that there was an overrepresentation of higher intracranial pressure in our ME-CFS patients compared with what was expected in healthy controls. And this is something that we are following up on right now, together with the biomarkers. We are also doing a big study where we have temporary solved sampling from ME patients, and this is a collaboration between Sweden and Australia. And we are doing metabolomic analysis then, and I've been collecting over 3,000 samples in Sweden, doing home visits to patients and then monitoring them over time during many hours, during different activities like a mental activity, a social activity, and a physical activity. And my colleague Chris Armstrong in Australia has done something similar and is now collecting patients over several hours, but then looking at the influence of a standardized meal that is taken at given time points. And then we draw temporary solved small miniature volumes of blood from the patients to monitor the metabolome. And the metabolome is going to be measured by nuclear magnetic resonance, NMR, and with mass spectrometry. That is the technique that I'm using. And then we're going to integrate that and look at the variability over time in patients both in Sweden and Australia. And just some preliminary data from what we have done in our patients in Sweden. And this is a very, very small part of all the data we are collecting. We are going to monitor about over 800 different markers when it comes to metabolome. But we just looked at the lactate concentration. And lactate concentration is a result of a mitochondrial activity you get when you do some exercise or physical or mental strain activity. And as you see, many of the ME patients, which are the light blue bars here, they start slightly higher than our controls, or they are at resting state when it comes to lactate. After a small activity and exercise, they increase and they stay, as you see, over time, elevated. They don't decrease and they don't consume their lactate as they supposed to do. In a healthy control, you see that you produce some lactate, but you typically, with this light exercise, you typically don't get an elevation, or you break down your lactate actually based on your low activity exercise. So this is a simple but very significant finding, and we see this in both our ME patients and in rather many of our post-COVID patients, as you may understand. So that was my first part of my presentation. I'm also happy now to take some questions, I guess.
Der kun kommet there's only one question, and this is Line, who would like to know if you could share your reference for ME being twice as common as sclerosing or MS, because that's new to me too. That's a pretty simple question. You probably don't have the reference here, but if you could share it with us afterwards, that would be nice.
Yeah, I can share that. It's an old reference actually that has been but it's rather well known now. And of course, we know that there is an underrepresentation of diagnosis in ME and maybe less underdiagnosis in MS. But still, ME is twice as common as MS.
Yeah.
In our populations.
Yeah, the Nordic population.
Yeah. And also in United States also actually.
Okay. And also something about, could you elaborate on the parameters on the x-axis?
P-value means if it's significant or not. So this difference here is not significant. But this is below 0.001, so that's strongly significant, that change. So that is just the p-value. That is the significance threshold we have.
And did you repeat it, or there's these different?
Yeah, so this is some of many patients that have been monitored and many controls. So it's about 50 patients and 50 controls in each, and that gives the spread. So here's the mean value, and this is the range, basically. So this is, of course, not. I mean, as you understand, some patients are actually rather low, and some patients are very high. But when you do this median mean value distribution, you see that they are, in this case, elevated, although not significant. Here's very significantly elevated. Here, very significantly elevated, and also here.
But is it the same patient who's going to different activities, or? Yes,
but in this case, I just show you one example. This is a slight exercise by sitting on an ergonometry bike. So they actually are just, it's like the exercise is it resembles walking slowly, maybe a few hundred meters. So it's really not heavy exercise.
Thank you. There is another question here from a member. Is it only neurotransmitters that are infected, or is it also the neurons themselves? Do they die, or what happens with them? Normally, dead neurons are gone for good. So, could you explain?
That's a good question. I mean, we think we see that there is both effects on the neurotransmission itself, the signaling, but also that neurons get affected and get degraded. So you have a neurodegenerative loss, basically. That, of course, is not a good thing. It is, to a large extent, not reversible, so you cannot really grow new neurons everywhere. We have in the human adult brain still the plasticity. So actually we can get other neurons to take the role of a dead or lost neuron. So we can retrain the brain to actually cope with the situation to some extent. But inflammation and damages to the central nervous system are to a large extent problematic because they are not reversible in the same way as a damage in a peripheral organ and so on.
Yeah, thank you.
And another thing that we are concerned about is that once you have an inflammation, even if you don't totally destroy a region of the brain or some neurons, you can get scar tissue formation. So you will have configurations of the brain tissue that makes the immune system alerted, and that the immune system will try to attack that part of the brain and then cause further damages, unfortunately.
Thank you. Next question is from a patient here who has ME-like symptoms following the corona vaccination. So hopefully would your research also cover not only post-COVID but also post-vaccine disease?
Yeah, of course we are also interested in to monitor that. We don't see that many reports on that in our cohorts, but of course any kind of medication or any kind of vaccination is of course also the whole principle of vaccination is to have a provocation of the immune system to give us a protection and hopefully save us from severe disease. But as we know, some patients have been getting worse by vaccination or immune activation. So you cannot rule that out totally. The thing we know is that most patients are protected against severe illness, severe critical illness by being vaccinated. But even if you are vaccinated, you still can get COVID infection. Maybe a bit milder, but you still can get it. So one consequence that we have been looking at a bit is that if you are unlucky and maybe you have, without having so much symptoms, maybe been infected by COVID, and then you go and take your vaccination at the same time point, maybe that combination, being infected and having a boost of your immune system, could be negative or give a higher risk to actually give you consequences afterwards. We don't know for sure, but that could be a possible explanation.
Yes, thank you. Next question is from a patient with ME here who claims that you can recover with training your brain. Is that a go-to way, in your opinion? Training
training the brain is, of course, it's always good to stimulate your brain to some extent. But if you are critical ill, or if you have, for instance, ME, many ME patients have a very minimal possibility to activate, and many get instead post-exertional malaise problems. So it's a very delicate balance, and it's also a very personal set level of what kind of activation you should do or you could do without having a crash. And that comes both to mental and physical activation. So it's something that is, it's hard to say that what is the optimal window of that. It's very much up to yourself to feel what is possible and what is not possible. And what our recommendations is, of course, to try to avoid crashing, because crashes leads to longer time periods of severe symptoms and also may delay recovery and so on.
Okay, thank you. Last question. You talked at one of your slides about how the pressure on the optical nerve due to neuroinflammation is a way to measure this? Is it only, could you elaborate on this pressure on the optical nerve? Could it be due to something else than neuroinflammation, or how? Why is this important?
Yeah, this we still don't know exactly what is overpressure that we can measure by this ultrasound device, and we also can see in MRI, actually, when we do MRI on the patients sometimes, what that stands for. It could be a part of the inflammation. It could also be a part of circulation disturbances. The Bergquist clinic in Stockholm, for instance, they are very much interested in issues with the neck region of patients, and the effect of the problem with circulation in the neck region that could, in principle, also affect the central part of the brain, intracranial pressure, and that could be one part of the story also. So we still are interested to look into this. An interesting finding that we had was that after lumbar puncture, when you actually reduce the pressure partially during a very short time period, quite a few patients actually reported that they suddenly felt a bit less symptomatic. They had less brain fog and so on. But of course, that's not a long-lasting effect.
Can you tell how this is? Now we're talking about the CCI in the neck. How do you examine this? Is it an MIR scan, or is it a moving scan, or how do you check for that?
Yeah, so it's MRI scans typically, and of course there have been discussions on if you should have a standing up MRI, or as the common one is laying down. Currently we don't have opportunity to do standing MRI, but still finds if there is disturbances in circulation, and also problem with circulation. Basically, you can see that in the MRI camera. Not in all patients, of course, but in quite a few. Yeah. And I will, in my second part, I will talk more about the MRI and imaging, what we are currently developing. So maybe we can, leave some questions for that also.
Okay. Also, will you get into SPECT scanning? Is it something you do, or you don't have that possibility?
We have the possibility. We haven't seen so much in that yet of usefulness, but it's a good complement. PET imaging, of course, is a fantastic sensitive technique, but much more invasive, and nothing you can repeat over time. That's why I think MRI is going to be very advantageous. It's a simple examination, it's non-invasive, and it's rather it's less expensive, let's say, at least compared with other techniques.
Thank you. Last question before we go to a break. What is the possibility of being cured for ME? Is it impossible, or which group of patients do you see has better options for getting better?
Very good question also. I've been over the years trying to collect reports from patients who have been who has recovered fully or to a large extent, and I have actually managed to find those patients. It's not so easy, because once you're cured and you don't want to maybe associate yourself with the disease anymore, you're not in the records, you're not in the healthcare system anymore, so it's hard to find patients that fully recovered. But I have managed to get in contact with quite a few through different medias, and it's of course, it's if you want to have some general terms of why you have a higher likelihood of getting fully recovered, age is one thing for sure. The younger you are, the more likely you will heal. That is common for any other disease also, so it's not specific for ME, for sure. But I think other common features are that the patient has handled their disease in a good manner. They have accepted the disease in some way, but also been very careful with their energy levels, and careful with not over exercising and over training, to let it take some time to heal. And one very important factor, as we have seen, is also how they have been treated by healthcare or society and family, etc. So how the surrounding around the patient has been involved, and what kind of support and understanding the patient has received, has been extremely important for getting better also.
Yes. Thank you. So now we will take a break.